Hi everyone, welcome back to the Science Box. So today I'm going to be telling you everything you need to know for the cell structure chapter of AQA A Level Biology. The timeline's in the description, so if there's a particular part you're stuck on, feel free to just skip to there. The first part of this chapter is about microscopes. When dealing with microscopes, you may be asked to calculate the real size of the object you're looking at, and you can do that with the equation magnification equals size of image over size of object. If we need to find our magnification to do this, we'd multiply the eyepiece lens magnification by the objective lens magnification. People often confuse the resolution of a microscope and the magnification. Resolution is the minimum distance apart the two objects can be in order for them to appear as separate items. Increasing magnification increases size of image, but not always resolution, because every microscope has a limit of resolution. The disadvantage to light microscopes is that they use light rays, which have a relatively long wavelength. Electrons have shorter wavelengths, so can distinguish between objects closer together. So electron microscopes have a higher resolution than light microscopes. Just for comparison's sake, light microscope can distinguish different objects with a minimum distance of 0.2 micrometers, and certain electron microscopes can distinguish different objects with a minimum distance of 0.2 nanometers. The defining feature of an electron microscope is that it uses a beam of electrons. There are two main advantages to this. As we said, this gives it high resolving power because electrons have a shorter wavelength, and the beam can be focused using electron magnets. The two types of electron microscopes are transmission electron microscopes, or TEM, and a scanning electron microscope, or an SEM. We'll start by looking at a transmission electron microscope. The TEM consists of an electron gun that produces a beam of electrons that is focused onto the specimen by a condenser electromagnet. The beam passes through a thin section of the specimen. Parts of the specimen absorb electrons and therefore appear dark. Other parts of the specimen allow electrons to pass through, so appear bright. The resolving power of a TEM is 0.2 nanometers, but this cannot always be achieved because difficulties preparing the specimen limit the resolution that can be achieved, and a higher energy electron beam is required, and this may destroy the specimen. The TEM does have some limitations. Firstly, the whole system must be in a vacuum, therefore living specimens cannot be observed. Also, a complex staining process is required, and the specimen must be extremely thin. The image may contain artifacts, which are basically mistakes like air bubbles and things visible that aren't the specimen itself and shouldn't be there. And finally, the image is 2D. With the scanning electron microscope, the specimen does not need to be thin as electrons don't penetrate. The SEM directs a beam of electrons onto the surface of the specimen from above rather than penetrating it from below. The beam passes back and forth across a portion of the specimen in a regular pattern. The electrons are scattered by the specimen, and the pattern of this scattering depends on the contours of the specimen's surface. The resolving power of the SEM is 20 nanometers, so lower than the TEM, but the SEM can build up a 3D image with the help of a computer. We can use electron microscopes to look up close at organelles. To do this, we first prepare the organelles in a process called cell fractionation. Cell fractionation is the process where cells are broken up and the different organelles they contain are separated out. Before cell fractionation can begin, the tissue must be placed in a cold, buffered solution of the same water potential as the tissue. It needs to be cold to reduce enzyme activity that might break down the organelles. It needs to be buffered so that the pH does not fluctuate. Any change in pH could alter the structure of the organelles or affect the functioning of enzymes. and it must be the same water potential to prevent organelles bursting or shrinking as a result of the osmotic gain or loss of water. There are two stages to cell fractionation. Homogenation is where cells are broken up by a homogenizer or blender to release organelles from the cell. The resultant fluid, known as the homogenate, is then filtered to remove any complete cells and large pieces of debris. Ultracentrifugation is the process by which the fragments in the filtered homogenate are separated in a machine called a centrifuge. This spins tubes of homogenate at very high speeds in order to create a centrifugal force. The process for animal cells is the tube of filtrate is placed in the centrifuge and spun at a slow speed. The heaviest organelles, which would be nuclei, are forced to the bottom of the tube, where they then form a thin sediment or pellet. The fluid at the top, called the supernatant, is removed, leaving just the sediment of the nuclei. The supernatant is transferred to another tube and spun in the centrifuge at a faster speed than before, so the next heaviest organelles, the mitochondria, are forced to the bottom of the tube. The process continues, so at each increase in speed, the next heaviest organelle is sedimented and separated out. Now moving on to eukaryotic cells and their ultrastructure. The ultrastructure is the internal structure of the cell. Let's start with the nucleus. The nucleus is the most prominent feature of a eukaryotic cell. It contains the organism's hereditary material and controls the cell's activities. The parts of the nucleus are the nuclear envelope, which is the double membrane that surrounds the nucleus. The outer membrane is continuous with the endoplasmic reticulum of the cell and often has ribosomes on its surface. It controls the entry and exit of materials in and out of the nucleus and contains the reactions taking place within it. The nuclear pores allow the passage of large molecules in and out of the nucleus. For example, mRNA travels out of the nucleus through nuclear pores. The nucleoplasm is the granular jelly-like material that makes up the bulk of the nucleus. There are chromosomes, which are protein-bound linear DNA, and the nucleolus, which is a small spherical region within the nucleoplasm. It manufactures ribosomal RNA and assembles the ribosomes. The functions of the nucleus are: it acts as a control center of the cell through production of mRNA and tRNA, and hence protein synthesis. To retain the genetic material of the cell in the form of DNA and chromosomes, and to manufacture ribosomal RNA and ribosomes. With mitochondria, around the organelle is a double membrane that controls the entry and exit of material. The inner of the two membranes is folded to form extensions known as cristae. Cristae provide a large surface area for the attachment of enzymes and other proteins involved in respiration. The matrix contains proteins, lipids, ribosomes, and DNA that allow mitochondria to control the production of some of their own proteins. Many enzymes involved in respiration are found in the matrix. The function of the mitochondria is, firstly, it's the site of the aerobic stages of respiration. And leading on from this, it's responsible for the production of the energy carrier molecule ATP. Okay, now moving on to chloroplasts. So first we have the chloroplast envelope, which is the double plasma membrane that surrounds the organelle. It's highly selective in what it allows to enter and leave the chloroplast. The grana are the stacks of disc-like structures, and these discs are called thylakoids. Within the thylakoids is the photosynthetic pigment chlorophyll. Grana are where the first stage of photosynthesis takes place. The stroma is the fluid-filled matrix where the second stage of photosynthesis takes place. Within the stroma are a number of other structures such as starch grains. Here are the adaptations of chloroplasts. The granule membranes provide a large surface area for the attachment of chlorophyll molecules. The fluid of the stroma possesses all enzymes needed to make sugars in the second stage of photosynthesis. The chloroplasts contain both DNA and ribosomes, so they can quickly and easily manufacture some of the proteins needed for photosynthesis. With the rough endoplasmic reticulum, ribosomes are present on the outer surface of the membrane. It provides a large surface area for the synthesis of proteins and glycoproteins, and it provides a pathway for the transport of materials, especially proteins, throughout the cell. The smooth endoplasmic reticulum synthesizes, stores, and transports lipids and carbohydrates. The Golgi apparatus consists of a stack of membranes that make up flattened sacs, or cisternae, with small, rounded, hollow structures called vesicles. Proteins and lipids pass through the Golgi apparatus in strict sequence. The Golgi modifies these proteins, often adding non-protein components, for example carbohydrates, to them. It also labels them, allowing them to be accurately sorted and sent to their correct destinations. Once sorted, the modified proteins and lipids are transported in Golgi vesicles. The functions of the Golgi are to add carbohydrates to proteins to form glycoproteins, to produce secretory enzymes, for example those secreted by the pancreas, to secrete carbohydrates, for example those used in making cell walls in plants, to transport, modify and store lipids, and to form lysosomes. Lysosomes are formed when the vesicles produced by the Golgi apparatus contain enzymes, for example proteases and lipases. The functions of lysosomes are to hydrolyze material ingested by phagocytic cells like white blood cells and bacteria, to release enzymes to the outside of the cell in order to destroy material around the cell, to digest worn-out organelles so that the useful chemicals they're made of can be reused, and to completely break down the cells after they have died in a process called autolysis. Ribosomes are small cytoplasmic granules found in all cells. 80S ribosomes are found in eukaryotic cells, and 70S ribosomes are found in prokaryotic cells, mitochondria, and chloroplasts. Ribosomes consist of a large subunit and a small subunit, each of which contain ribosomal RNA and protein. The cell wall consists of microfibrils of cellulose embedded in a matrix. Cellulose microfibrils are strong. Cell walls contain a number of polysaccharides. The middle lamellae marks the boundary between adjacent cell walls and cements adjacent cells together. The functions are to provide mechanical strength to prevent cells bursting under pressure created by the osmotic entry of water, to give mechanical strength to the plant as a whole, to allow water to pass along it and so contribute to the movement of water throughout the plant. Vacuoles are fluid-filled sacs bounded by a single membrane. The single membrane around the vacuole is the tonoplast. Plant vacuoles contain solutions of mineral salts, sugars, amino acids, waste, and sometimes pigments. Here are the plant vacuole functions. To support the herbaceous plants by making the cells turgid. It contains sugars and amino acids that may act as a temporary food store, and the pigment may colour petals to attract pollinating insects. Now, you may be asked to compare and contrast prokaryotic and eukaryotic cells, so I've created a table of comparisons for you. Prokaryotic cells have no true nucleus, as the DNA is free in the cytoplasm, but eukaryotic cells have a distinct nucleus with a nuclear envelope. In prokaryotic cells, the DNA is not associated with proteins, but in eukaryotic cells, the DNA is. In prokaryotic cells, some DNA may be in the form of circular strands called plasmids. Plasmids reproduce themselves independently and possess genes that may aid the survival of the bacteria in adverse conditions. Eukaryotic cells have no plasmids and the DNA is linear. Prokaryotic cells have no membrane-bound organelles, but eukaryotic cells do. Prokaryotic cells have no chloroplasts, but both plant and algal cells have chloroplasts, and these are eukaryotic cells. Prokaryotic cells have smaller 70S ribosomes, but eukaryotic cells have larger 80S ribosomes. In prokaryotic cells, the cell wall is made of murin, which is a peptidoglycan, and in eukaryotic cells, where present, the cell wall is made mostly of cellulose, or chitin in fungi. Prokaryotic cells have an outer layer called the capsule, and eukaryotic cells have no capsule. Now moving on to viruses. Viruses are acellular, non-living particles. They are smaller than bacteria and contain nucleic acids, so either DNA or RNA, as genetic material, but can only multiply inside living host cells. The parts of a viral particle are attachment proteins, a lipid envelope, the matrix, the capsid, the genetic material, so either DNA or RNA, and reverse transcriptase, which is an enzyme that transcribes the viral RNA into DNA to be inserted into the host genome. Now let's talk about mitosis. Mitosis is needed for growth, repair, and reproduction. It produces two daughter cells that have the same number of chromosomes as the parent cell and each other. Before mitosis is interphase, where the DNA replicates and the two copies are joined at the centromere. Stage 1 of mitosis is prophase. The chromosomes condense and therefore become visible as they are shorter and thicker. Animal cells have centrioles which move to the opposite poles of the cell. The nucleus disappears and the nuclear envelope breaks down, leaving chromosomes free in the cytoplasm. The chromosomes are made up of two sister chromatids, which are joined at the centromere. In metaphase, the spindle apparatus developed from the centrioles attaches to the centromere of the chromosomes and pulls them to the equator of the cell. In anaphase, the centromere divides into two and the spindle fibres pull the sister chromatids to opposite poles of the cell. From this point, the chromatids are classed as chromosomes because they've separated. In telophase, the chromosomes decondense, becoming longer and thinner, leaving only chromatin. The spindle fibres disintegrate and the nuclear envelope and nucleolus reform. And finally, in cytokinesis, the cytoplasm divides. Here is the graph of how the mass of one cell and its DNA changes over the cell cycle. See if you can figure out at how many hours each stage takes place. Required Practical 2 is all about mitosis. It's the root tip squash experiment, which hopefully you will have done or will be doing in class. Step 1. Using a scalpel, cut off the tip of the root of an onion or garlic, 5 mm from the end of the root. This part of the root is where growth happens the fastest, so we are most likely to see mitosis in this area. Step 2. Place your root tip in a test tube along with hydrochloric acid and put it into a water bath for 10 minutes. The acid, along with the heat, softens the cells and loosens the root tissue. The acid also stops mitosis from continuing, so we've kind of frozen that process. Rinse the root tip with distilled water and place it onto a microscope slide. You can then use filter paper to soak up any excess water. Add 2 drops of Orsine stain to the root tip. This stain is like a purpley red colour. Lower on the cover slip with the mounted needle, avoiding air bubble formation, and leave the slide for 10 minutes, allowing the auxine stain to stain the chromosomes. Finally, place a piece of filter paper on the cover slip and press down gently. This is called macerating the cells, which basically means squashing them and breaking up the root tip. This makes it a single layer of cells, so light can pass through when viewing the slide through the microscope. Then you can view the slide through the microscope. You may be asked to calculate a mitotic index from this, which is the number of cells undergoing mitosis divided by the total number of cells, all multiplied by 100. Prokaryotes don't use mitosis to divide, they use binary fission. Step 1. The circular DNA replicates and both copies attach to the cell membrane. Step 2. The plasmids replicate. Step 3. The cell membrane begins to grow between the two DNA molecules and begins to pinch inwards, dividing the cytoplasm in two. Step 4. New cell walls form between the two molecules of DNA, dividing the original cell into two identical daughter cells. Both will have one copy of the circular DNA and a variable number of plasmids. Viruses replicate by first attaching to the host cell with their attachment proteins. They then inject their nucleic acid into the host cell, and the viral nucleic acid is coded for by the host cell, so the new virus particles are assembled. Cancer is due to the damage to the genes that regulate mitosis. This creates uncontrolled growth. Malignant tumours grow rapidly, are less compact, and more likely to be life-threatening. Benign tumours grow slowly, are more compact, and are less likely to be life-threatening. Cancer can be treated by blocking parts of the cell cycle. Therefore, cell division and cancer growth stops. Drugs used usually either prevent the DNA from replicating or inhibit metaphase by interfering with spindle formation. These drugs also disrupt the cell cycle of normal cells, which is a disadvantage, but the drugs are more effective against rapidly dividing cells. Hair-producing cells divide rapidly, and therefore these drugs usually result in hair loss. Thank you for watching. Please like and subscribe to follow the rest of our teaching videos. Follow us on socials to get some quick-fire content so that you don't have to feel guilty about that doom scroll. And finally, there's a link in the description to all the academic help we offer here at the Science Box. Thank you.